Tonight, we're going to be looking at photosynthesis. I'm calling it Photosynthesis Solar Power for Life. My presentation will last about an hour. We will have a video kind of in the middle of it. And the video to me is kind of a highlight. It's high school biology class on photosynthesis, and I think it's an outstanding video myself. Then we'll have time for a Q&A after that, and I think we'll be done by about 9, but we'll see. Next month, Krista Bontrager, one of the RTB staffers, will be joining us, and she'll be presenting on finding Christian unity in creation. Some of you may have tuned into her RTB Live presentation that she gave in July, I think it was. But anyway, this is essentially that same presentation, but she's going to be giving it to us live, and she will be answering our questions following the presentation. We don't meet in December's, and because of Christmas and all the other general conflicts, maybe that's not as much of an issue this year with the COVID virus, but anyway, at this time, we're not planning to meet in December. Next week, there's a conference on apologetics, Christian apologetics, called Hold Fast, and it's a virtual live-streamed conference. It's actually in the evenings. It doesn't start until 7 o'clock Eastern time, which basically is 4 o'clock our time, and then goes on for a couple, 2 or 3 hours, I think, just in the afternoons. You do have to sign up for it. You can't just click onto it. It's presented by the Southern Evangelical Seminary, and there's a number of RTB speakers, and I'm showing a few of the speakers here, but there's quite a few others as well. Now that we're into the COVID virus, and many of you are spending more time at home, I want to suggest that you take in some RTB reading. And so I've got a list of books that I'm suggesting to you. If you've tuned into our meetings before, you know that I've already been pushing these books on our meetings. But the first one is Classic Christian Thinkers by Ken Samples. And he talks about some of the early church fathers: Athanasius, Augustine, Thomas Aquinas, Martin Luther, and C.S. Lewis. This book was released in July of last year. It's a joint effort between RTB scholars Fuzz Rana and Ken Samples, and it shows that great strides are being made in human enhancement with gene editing techniques, brain-computer interfaces, prosthetics, and anti-aging technologies. And it's basically, the idea is transhumanism, and the idea is to transform humans through science and technology to elevate us to an evolutionary post-human step. Now, whether you or not you think that there's any validity to this idea, it is being pursued on many fronts right now. So this is really kind of an opportunity to see what's happening in this field, including the ethical ramifications of it, which are not inconsequential. This is Jeff Zwering's new book called Escaping the Beginning, Confronting Challenges to the Universe's Origins. And Jeff considers in this book the most prevalent of the eternal universe theories: quantum gravity, the steady state model, the oscillating universe, and multiverse theories, and the implications of these theories for the existence of a beginner. Weathering Climate Change is Dr. Ross's latest book, and it was released this spring. And for those of you who had tuned into our meeting last month, this was the subject of our meeting, and Dr. Ross joined us at that time. So this won't be new news to many of you, but if you didn't get a chance to tune into that, this is a great book. He points out that many factors besides carbon dioxide levels influence climate change, and he accepts that global warming is real and humans have contributed to it, but he says that neither denying the reality of climate change or insisting on draconian remedies makes a whole lot of sense. And instead, he suggests that there are win-win solutions which can mitigate the negative effects of global warming while avoiding negative economic and social impacts. Then there's a couple of conversation cards, which Reasons to Believe is released within the last couple of years. This first one has you see 4 panels here, and I think there's 4 more. So it's double-sided, and it folds up neatly, and it would be something that you could hand to friends that you want to introduce to the gospel. This basically is this particular card is talking about Reasons to Believe's testable creation model. And then there's another card which is focusing more on the Big Bang and how the Big Bang Big Bang cosmology ties into the book of Genesis, and particularly the rest of the books of Scripture. So, and then there's a video which has recently been released called The Heart and Mind Behind Rational Christians, and this was prepared by the 4 scholars, Hugh, Fuzz, Jeff, and Ken, and it tells how each one of them came to know the Lord and came to faith and ultimately to ministry at RTB. And that's all of the books that I have now, but I do want to point out that if you can find any of these books at the reasons.org website, you will see a store icon, and you will also see a donate icon. If you go to the donate icon, you will see that for a donation of any amount, you can get a copy of Fuzz Rana's brand new book, which hasn't even been released yet, called Thinking About Evolution. Frankly, I'm not sure when it's going to be released. It was supposed to be released around the 1st of October, but because of the coronavirus, it's been delayed at least through the end of October and maybe into November. But for a donation of any amount, you'll be on the waiting list as soon as it is released. So our talk tonight is photosynthesis, and we're going to include in the talk of photosynthesis also the chloroplast. In fact, most of the discussion will be on the chloroplast, but this is actually part of a 3-part series or trilogy in which I described ribosomes in the first part, mitochondria in the second part, and in this one we'll talk about photosynthesis. We'll see how photosynthesis completes the cycle, transforming sunlight into oxygen and ATP and converting carbon dioxide into the carbohydrates, specifically the sugars that the mitochondria in our bodies use. And all 3 of these presentations, I think, provide compelling evidence for fine-tuning and intelligent design and give us more reasons to believe in the creative God of the Bible. Just a quick review of the other 2 presentations. In the ribosome presentation, I pointed out that ribosomes manufacture all of the proteins used by all life forms. All life forms have ribosomes, and they take DNA, which are transcribed into messenger RNA, and then the mRNA is translated using the genetic code into the amino acids, which the ribosome then strings together into proteins. Even though the chloroplast is the ultimate source of all of our energy, we don't have chloroplasts in our body. We have mitochondria. And mitochondria take the glucose in our body, which is actually ultimately produced by photosynthesis, but then we eat foods that contain glucose, and the oxygen we breathe and converts them into ATP in a process called respiration, giving off carbon dioxide as a waste product. And we will see that plants use sunlight, water, and carbon dioxide to manufacture oxygen, ATP, and glucose, forming the basis of the food chain. The glucose produced by photosynthesis is at the base of the food chain. The origin of photosynthesis traces back nearly to the origin of life on Earth more than 3.5 billion years ago. Photosynthesis manufactures about 175 billion tons of carbohydrates each year. And carbon dioxide is a potent greenhouse gas, so by producing oxygen and converting carbon dioxide into food, photosynthesis is doing its part to help us fight global warming. I had a lot of fun putting this presentation together because as I got deeper and deeper into it, I kept discovering more and more complicated and fascinating features of the process of photosynthesis, which seemed to me to defy evolutionary explanations. It was kind of like peeling an onion. When you look at an onion, all you see is the outer skin, but as you peel the skin, you come to another layer and then another layer and so on, revealing more and more about the onion until you finally reach the onion's core. I don't think I even got close to the core of photosynthesis. Tonight, we'll think about the origin of photosynthesis, which dates back nearly to the origin of life itself, and we'll watch a video on how the chloroplast performs photosynthesis, which is pretty much the same way that cyanobacteria does, but we'll look at how the watchmaker argument, which we talked about in both the ribosome and mitochondria presentations, apply also to photosynthesis, and we'll consider some recent scientific evidence supporting the idea that photosynthesis provides strong evidence for intelligent design. This is a picture of a microscopic organism called cyanobacteria. They're thought to be among the very earliest organisms on Earth, and they date back to around 3.5 billion years ago, or maybe even earlier. There's some reason to believe that possibly this could go back as far as the first, or very shortly after the first life was observed, as much as 3.85 billion years ago. And that's right after the late heavy bombardment, which basically means during the late heavy bombardment, the Earth's surface was molten. So any life existing before that or during that time would have been very problematic, I think. There are several unique things about cyanobacteria. First, they're photosynthetic like chloroplasts, and that is they convert sunlight, water, and carbon dioxide into energy. Second, unlike most other bacteria, they form colonies like those shown here. So most other bacteria kind of float around singly, but these things link to each other in these chains. And finally, they have left a fossil record supporting the notion that they existed on Earth almost from the beginning of life. Here's another picture of cyanobacteria, again, just showing a slightly different setting, but again, showing that they're connected in these chains. Cyanobacteria form in mats on structures like these, which are called stromatolites. These exist in several places around the world, particularly in Greenland and Australia. These particular ones are in Australia. Even though cyanobacteria are microscopic, these mats build up over long periods of time, and kind of like coral, the live cyanobacteria exist on the top of the stromatolites over the fossils of their ancestors. So we'll take a picture, we'll take a look at the fossils in a moment. But here's a picture of stromatolites showing that they are in fact underwater, but in shallow water. Here's a cross section of a stromatolite showing the layers of cyanobacterial fossils. As I said, the evidence says they go back to at least 3.5 billion years ago. The earliest life form is thought to have been bacterial, though not photosynthetic. But there's evidence that it dates back to about 3.85 billion years ago, as I said earlier. However, there's also evidence that a limited amount of free oxygen was present on Earth that long ago. Here's a slide that Hugh Ross often uses in his presentation, showing that there was a little bit of oxygen present in the atmosphere from the earliest days of life. And the RTB theory is that cyanobacteria must have been present nearly as soon as the first bacterial life. But this would be remarkable since cyanobacteria are about 10 times as complex as regular non-photosynthetic bacteria. The reason there was so little atmospheric oxygen is that the oxygen that was available got taken up almost immediately to oxidize the pure metals that were present on early Earth. We discussed the endosymbiotic hypothesis during my previous presentation on the mitochondria. Like the mitochondria, which most biologists believe evolved after a larger cell consumed an alpha protobacteria, the chloroplast is assumed to have evolved from a similar event or series of events. Chloroplasts are believed by most biologists to have developed from symbiotic cyanobacteria. the theory states that a prokaryotic bacterial cell was consumed by a larger cell, but somehow was not digested. Later, both cells developed a mutualism, and eventually the chloroplast evolved into the organelle, which provides photosynthesis for plant cells. Here we have a modern cell in a plant leaf. Note that this cell contains 5 chloroplasts. Hold on just a second. Let me see if I can show you this. These are the chloroplasts here. If you see this laser, these are the yellow and green objects. There's 5 of them shown here. And but typically there are often 100 or more chloroplasts in each cell of plant leaves. Only 5 are shown in this particular one. Of course, this is a cross section. We don't know how deep this is. Plant cells also contain mitochondria, typically 300 or 400 of them, only 100 or so chloroplasts, but 300 or 400 mitochondria. Here's a mitochondria. Here's one here. And they also originated supposedly in early plant cells from a separate endosymbiosis. The cell also contains a nucleus, that's here, with chromosomes containing DNA, as well as ribosomes. It shows a ribosome here. I think the ribosomes typically are actually on the endoplasmic reticulum here, but this kind of shows that they're free floating in the plant cell. I don't know that that's correct, but that was the best plant cell I could find. So that's what you see is what you get. The nucleus contains chromosomes containing DNA, and then the ribosomes, of course, as I said. The chloroplasts and mitochondria, thought to have originated from bacterial cells, also have nuclei, DNA, and ribosomes, and they manufacture some of their own proteins. But as we'll see, most of the proteins in both the chloroplast and mitochondria are actually manufactured here from the nuclear DNA by the cell's ribosomes, not by the chloroplast DNA and ribosomes. Then they're transported into the chloroplast and mitochondria. We'll talk more about this later, but now let's take a close-up look at a chloroplast. This is a cartoon of a chloroplast. The chloroplast, we'll see more about the details of the chloroplast here, but the main reason I wanted to show you this is that the chloroplast also has DNA and ribosomes. So here's some of the DNA. You can just barely see it. It looks kind of like a purple squiggly. Here's another little bit of it. And then there's also all these ribosomes, these black dots. And that indicates that the chloroplast does make some of its own proteins. But the important point here is that most of the chloroplast proteins are made in the plant cell, and then they have to be transported to the chloroplast. Remember that I said there's about 100 chloroplasts in each plant cell. It's also interesting, I think, that parts of the protein complexes in the chloroplast are made in the plant cell, and parts are made in the chloroplast. Generally speaking, when there's a protein complex in the chloroplast, it's actually some of it's made in the nucleus and some of it's made in the chloroplast itself, and the larger pieces tend to be made in the chloroplast. So it's important to distinguish between nuclear DNA, which is in the cell, and chloroplast DNA, which is in the chloroplast, and we'll talk more about this later. I don't want to make fun of the endosymbiotic hypothesis since it holds sway with a large majority of evolutionary biologists. Nevertheless, there are some obvious problems with the hypothesis, 3 of which I've listed here. First, endocytosis, sometimes called phagocytosis, is generally the way single-celled organisms eat their prey. The outer membrane essentially engulfs or wraps around the prey, and it is absorbed into the host and digested. There's no known process by which in just one generation an endocytosis event could occur and a symbiotic relationship develop between the 2 organisms. This would normally take multiple generations and would require the first organism not to digest the second, which would be the usual outcome, which would be the end of the symbiotic relationship, of course. Second, over some period of time, the two organisms would have to adapt to each other symbiotically so that each gains some advantage. I have not seen an acceptable explanation for this. Finally, chloroplasts only have 170,000 to 200,000 DNA base pairs containing about 100 genes, whereas the smallest known cyanobacterial cell has several million DNA base pairs with 1,500 genes. Most of the chloroplast DNA functions actually occur in the nuclear DNA. This is usually explained as the effect of evolutionary processes over 1 billion or more years, where the overwhelming majority of the chloroplast DNA functions have become incorporated into the cell's nuclear DNA, leaving only a few basic functions to be performed by the chloroplast DNA itself. I don't find this evolutionary explanation to be very satisfying, frankly. Something like the endosymbiotic hypothesis, though, is necessary for scientists who reject supernatural explanations because of their commitment to methodological naturalism, which is an assumption of the scientific method which only accepts natural explanations for phenomena. But here's an alternate hypothesis. God created plants with a photosynthetic capability by adapting the successful design of cyanobacteria cells into chloroplasts found in complex plant cells, just as engineers routinely adapt successful existing designs into their inventions. This hypothesis assumes supernatural creation events, such as those spelled out in Genesis. Recent biology students at Beverly Hills High School, yeah, that's Beverly Hills in Hollywood, got to see the following video, which I thought gives an excellent explanation of the process of photosynthesis and the chloroplast.
So today's video is on photosynthesis, and it might seem pretty straightforward. You shine some sunlight, you sprinkle some rainwater, you add some carbon dioxide from the atmosphere, and through photosynthesis, plants will give off oxygen and will create simple sugars such as glucose. But the actual process is a lot more complex. So let's get started. So the sun gives off a variety of types of energy, and this energy travels across space in the form of a wave. And the titles of the energy you can see at the top of the picture. Now we're going to focus our attention on visible light because it's the wavelength of visible light that drives and powers photosynthesis. Well, let's focus on how light is absorbed, and when we do, we're going to see that the sun gives off visible light, also called white light. White light is actually a mixture of Roy G. Biv, and what this is are the colors of the rainbow: red, orange, yellow, green, blue, indigo, violet. All these colors are a mixture known as white light. Well, how do we know this? Because when you shine light into a prism, when you shine white light into a prism, a rainbow comes out the other end. Just like you can see in this animation. White light is entering from the left, and because of the curvature of the prism, Roy G. Biv, the colors of the rainbow are coming out the other end. Notice how the wavelengths are different. Red has a longer wavelength, violet has a more narrow. And so this is how we know that white light is made from a mixture of these colors. Here's an actual picture of white light entering a prism, and you can see on the left-hand side a rainbow is coming out the back end. Moisture in the air can act like a prism and create a rainbow. You ever notice after it rains, you can sometimes see a rainbow in the sky, or in this case, near a waterfall. Because of all the moisture in the sky, the moisture separates the sunlight, into the rainbow colors that you see. And so colors are either reflected or absorbed. When sunlight shines on an object, what we see is reflected light. The reason this leaf appears green is because it's reflecting the green wavelength of light. When light shines on these bananas, the reason it appears yellow is because the yellow wavelength reflects, while the other colors are absorbed. When we shine light on these balloons, the blue wavelength reflects into our eyes, and the other colors are absorbed. When we look at white objects like snow or this Arctic hare, the reason objects appear white is because white reflects all the colors and all the light, all the wavelengths of light enter our eyes, and our brain interprets this as the color white. Well, the opposite would be black. When light shines on a black object, nothing reflects. It all is absorbed, and that's why this automobile appears black, because there's no light reflecting into our eyes. So when we relate this to photosynthesis, plants have mostly green leaves, which means that they're going to reflect the green wavelength, but the other colors are absorbing into the leaves and are going to be powering photosynthesis. In this graph, we can see, for instance, that this graph shows the wavelengths of light that most power photosynthesis. Notice how there's a peak around 400 nanometers. Well, that's the wavelength of blue. There's another peak around 700 nanometers. Well, that's the wavelength of red. So blue light and red light are most stimulating for plants in the process of photosynthesis. Ironically, the color green around 550 nanometers, the color green that we most associate with plants, actually has the least amount of impact in their ability to do photosynthesis. That's because most green light is reflecting. Very little green light is actually absorbing into the leaf. So when we look at a general definition of photosynthesis, it's the process that converts solar energy into glucose. And this formula here outlines photosynthesis. And in the reactants, we have carbon dioxide and water, and in the presence of sunlight, we'll produce sugars such as glucose and oxygen. Well, who performs photosynthesis? Well, these are the autotrophs. Now, we typically think of plants as doing photosynthesis, but they're not the only ones. Algae, phytoplankton, the basis of ocean food webs, kelp. Kelp can grow into these enormous underwater forests. And even some bacteria known as cyanobacteria are photosynthetic. So there's a lot more autotrophs than just plants. So photosynthesis is also what we call an endergonic chemical reaction. And what that means is that photosynthesis requires the input of energy. From the picture, I'm implying that sunlight. Sunlight is the energy that is input to power photosynthesis. And when we talk about the end result, what's created? Well, oxygen, which is a waste gas, and simple sugars like glucose. But when we zoom on into the leaf, we find some peculiar pores on their underside. These pores will open and close, and they're called stomata, and it's how they exchange gases. It's how they take in carbon dioxide from the atmosphere, and when they produce oxygen as a waste, through these stomata openings, they give off the oxygen created. So it's through these pores that they exchange gas with the atmosphere. Okay, so now let's introduce the structure in cells that allows them to do photosynthesis, and this is the chloroplast. What we're going to do is we're actually going to divide photosynthesis into two stages. Stage one being the stage that depends upon sunlight, the light-dependent reactions. And these take place in the thylakoid membranes of a chloroplast. I want to actually look at a more simplified diagram here. And when we do, we can actually see the thylakoids, which kind of look like coins or frisbees stacked on top of one another. Well, in the membrane of these thylakoids is where the light-dependent reactions occur. And if you ever come across the term granum, that's a term that means a stack of thylakoids. Okay, so you see that flashing black box? I want to zoom into that area of the chloroplast. And when we do, we're going to see that there's some things actually embedded within the membrane of the thylakoid. I've labeled them PS1 and PS2, and these are the photosystems, and we're going to talk about them in more detail, but for now, in the photosystems is where we find chlorophyll and various protein molecules that are responsible for capturing and transferring energy during the light-dependent reactions. And it's during the light-dependent reactions where oxygen, ATP, and something called NADPH is created. We'll talk about these molecules in more detail coming up very shortly. And then we'll shift into stage 2, the light-independent reactions. And this happens or occurs in the stroma, the fluid-filled interior of the chloroplast, just outside of the thylakoid. And it's in stage 2, the light-independent reactions, where glucose is actually created. So let's talk about these 2 stages for the rest of this video. So now let's get into some of the details of the light-dependent reactions. And what we're going to do is we're going to break this down into a few steps. So step number 1, chlorophyll in photosystem number 2 will absorb sunlight. Now, even though it's called photosystem number 2, it's actually the first in the chain of events. They were named photosystem 1 and photosystem 2 by order in which they were discovered, not the order in which they perform their function. So you kind of have to get over that backwards name right there. So in my animation, sunlight is striking the chlorophyll molecules in photosystem number 2, and this causes their electrons to become excited. And what happens is the electrons actually flow into the thylakoid membrane. And so as these electrons from chlorophyll in photosystem number 2 flow through the thylakoid membrane, this makes the membrane negatively charged. And as the electrons are flowing through, there's also proteins embedded within the thylakoid membrane that are aiding their travel. I just didn't show them in my animation. But this starts what we call an electron transport chain. It's a chain of events set in motion by these electrons. So when we move on into the second step, I've added some hydrogen ions into the stroma because there's dissolved molecules and ions in the stroma, and these hydrogens are going to play an important role. And so what happens is, as the electrons move through the thylakoid membrane, they actually will operate in power and turn on these protein pumps that will pull the hydrogens into the thylakoids. And as more and more hydrogens accumulate, this is going to make a really large concentration of hydrogens, which we'll see why that's important in a little bit. I also want to mention that water molecules are broken. So here's a water molecule, and with the help of enzymes, the hydrogens are broken off, and the electron from the hydrogen went into photosystem number 2. Here we go again. Enzymes will break off the hydrogen, and notice how the electrons were pulled into photosystem number 2. You know, photosystem number 2 has been losing electrons, and so it's the hydrogen of water, it's their electrons that will replenish photosystem number 2 so it doesn't run out of electrons, and so this process can keep going. This is why you have to constantly water a plant. Also, oxygen. This is where the oxygen comes from. See that O for oxygen? It will bond with another O from another water that was broken to make O2. The oxygen that we breathe used to be attached to water, which I think is kind of interesting. So, as we move on into the next step, again, sunlight is striking photosystem number 2, and its electrons are moving through the thylakoid membrane. But you know, sunlight is also striking photosystem number 1, and photosystem number 1 is filled with chlorophyll, and it has electrons that also become excited and move through the thylakoid membrane. So what we really have is kind of a longer electron transport chain. So now that we have our full electron transport chain, let's move on to the next step. And in the next step, a molecule called NADPH will be created. So in the stroma, NADP and a hydrogen are drawn together. And the electrons of the electron transport chain help these two bond together to make a really important molecule called NADPH. NADPH can kind of be viewed as a carrier of hydrogen, like a taxi whose job it is to carry hydrogen from one place to another. We'll come back to it in a little bit, but it's really important that NADPH was just created. So at this stage, the hydrogen is really beginning to accumulate inside the thylakoid from the breaking of water, from the electron transport chain pulling in hydrogens from the outside. So by now, there's a really large concentration of hydrogens inside the thylakoid. So when we come to our next step, because there's such a large amount of them, the hydrogens are going to start to diffuse through that enzyme labeled ATP synthase. And so what hydrogen does from a high to low concentration, it'll diffuse through ATP synthase and will help to bond a molecule of ADP, adenosine diphosphate, with another phosphate. And when that happens, they bond to make a molecule of ATP. And so this ATP, along with the NADPH, are really the most important things that are created during the light-dependent reactions. We're going to see what's so important about them in just a little bit. And so to summarize the light-dependent reactions, oxygen, NADPH, and ATP have been created. You know, sunlight was taken in, water was taken in, and oxygen was released as a waste. And in the light-dependent reactions, NADPH and ATP have been created. I do want to note at this stage, no glucose has been created. So let's move on to the light-independent reactions. Okay, so the light-independent reactions, you know, this is also known as the Kelvin cycle, named after the scientist who helped to identify these steps. And so we're going to start breaking it down. First of all, there's a 5-carbon molecule by the name of RUBP, ribulose biphosphate. Now, it has more than just 5 carbons. You can see 12 hydrogens, 11 oxygens, and 2 phosphoruses. But for simplicity, I've only drawn the 5 carbons of RUBP. And what happens is RUBP bonds with a molecule of carbon dioxide. And when they bond, they make a very short-lived 6-carbon molecule. The 5 carbons from RUBP plus the 1 from carbon dioxide make this very short-lived 6-carbon molecule. So the reason this 6-carbon molecule is short-lived is we finally get to see the importance of ATP and NADPH, which were created during the light-dependent cycle. This 6-carbon molecule is going to be broken down by the energy contained within ATP and NADPH. And enzymes will help to break down ATP and NADPH to release their energy. And when the energy is released, that 6-carbon molecule is broken down into 2 molecules called phosphoglycerate. Each PGA, or phosphoglycerate, is 3 carbons in size. And by the way, the NADP and the ADP that are left over will be reused and recycled in the next light-dependent reaction. And so this happens multiple times. Here's another short-lived 6-carbon molecule broken down by NADPH and ATP to make 2 more molecules of phosphoglycerate. And here's another 6-carbon molecule broken down by another NADPH and another ATP to make 2 more molecules of the phosphoglycerates. So what happens now with these phosphoglycerates? Well, some of those 3-carbon phosphoglycerates are going to bond with one another, and with the help of enzymes, they're going to bond to make simple sugars, such as glucose. And that's the whole point of photosynthesis, is to make glucose. Now, not all the phosphoglycerates do this. Some of the phosphoglycerates are going to help to keep the cycle going. And so what happens is they're going to be broken down. They're going to be broken down with the help of ATP to keep the cycle going. And when ATP breaks some of these other phosphoglycerates down, they're broken down. And then enzymes help to build that same 5-carbon molecule we saw earlier, ribulose biphosphate. And now we're repeating the Kelvin cycle, the light-independent reactions. The process simply restarts because carbon dioxide will bond with this 5-carbon RUBP. And when they bond, they make that short-lived 6-carbon molecule that we mentioned earlier. And the process repeats itself. So to summarize the light-independent reactions, carbon dioxide, ATP, and NADPH are going to create glucose. So when carbon dioxide is added to the ATP and NADPH from the light-dependent reactions, with the help of enzymes, you have simple sugars such as glucose created.
In 1802, if you all are still with me, the Reverend William Paley published this book entitled Natural Theology. In it, he put forth this argument. You stumble on a watch in a field and you realize it has not always been there, in contrast with a rock, which very well may have been there forever. You see that the watch is intricately designed and elegantly made, and you conclude by an intelligent being. You realize that living creatures also appear intricately designed and elegantly made by an intelligent being, and further, they have the ability to reproduce themselves, a property which even the most beautifully constructed watch does not possess. It is reasonable to conclude then that living creatures are also designed by an intelligent being. Although it was possible even as early as 1802 to see elegance in the form of living creatures, not a lot of details were available, especially at the microscopic level. Many scientists dismissed Paley's argument because they believed humanly designed mechanisms were very different excuse me, from living creatures. This was true up through the middle of the 20th century. Today, however, biological science has progressed to the point where it is possible to make detailed comparisons between the parts that make up living creatures and humanly designed mechanisms, giving new legs to the watchmaker argument. So the first one that I want to talk about is the photovoltaic cell. And it's very similar to the chloroplast or to cyanobacteria in that a number of photovoltaic or solar cells electrically connected to each other and mounted in a support structure is called a photovoltaic module. An array of these modules forms the solar panels in common use today, and which some of you may have installed on your roof. Some materials exhibit a property known as photovoltaic effect that causes them to absorb photons of light and to release electrons. When these free electrons are captured, an electric current results that can be used as electricity. And I'm comparing the photovoltaic cell to the chloroplast. Both the chloroplast and the photovoltaic cell use photons from the sun to convert solar energy into a different form of energy, ATP for the chloroplast, and electricity for the photovoltaic cell. Both use controlled processes to perform their tasks. This is in contrast with uncontrolled processes such as a wildfire or rust forming on the surface of a metal object exposed to the elements. An array of photovoltaic cells form a solar panel. An array of chloroplasts form a leaf. But there's also differences between chloroplasts and photovoltaic cells, obviously. For example, the energy produced in a chloroplast is chemical energy in the form of ATP, and in addition, chloroplasts use the solar energy to dissociate water molecules to produce oxygen, which is pumped back into the atmosphere. The photovoltaic cell produces electrical energy in the form of an electric current, and the manufacture and installation of photovoltaic cells is also energy intensive, and the photovoltaic cell does not produce oxygen. And furthermore, the chloroplast in the Calvin cycle, the chloroplast converts carbon dioxide into glucose, which forms the base of the food chain. In the photovoltaic cell, it has nothing comparable to the Calvin cycle. And although these cells are praised by environmentalists because they don't produce carbon dioxide, they also don't convert carbon dioxide into anything. The second example I want to use is ATP synthase. That was the enzyme that in the video we saw at the top of the thylakoid membrane, which was resulted from the hydrogen molecules passing through it and creating ATP from it. And you'll see on this, and I'm going to have to show my pointer again here. You'll see the ATP, the ADP and phosphorus enter here and are transformed in ATP as hydrogens enter the the lumen of the thylakoid and go into the stroma. And this rotates the whole thing around. You can see the rotation here and the creation of ADP and ATP. It's basically just like a motor. So the similarities between an ATP synthase and electric motor The ATP synthase has many of the identical components of the electric motor. It has a rotor, a stator, and bushings, and it's powered by hydrogen ions, that is, an electrical flow, and the rotation produces the ATP molecules. An electric motor, on the other hand, has many of the same components as ATP synthase, and it converts electricity into rotary energy. But there's differences, of course, also in that ATP synthase is microscopic and the electric motor comes in many different sizes depending on its purpose. Here's where we begin to peel back the layers of the onion. I really liked the video on photosynthesis and kind of wish I'd gone to Beverly Hills High School, but that's another story. Because the video explains the overall process of photosynthesis simply and correctly. However, the process isn't actually quite as simple as the video implies. For instance, the video showed that sunlight strikes chlorophyll in the protein complex called photosystem II, dislodging electrons at the beginning of the electron transport chain, and also dissociates a water molecule into 2 protons and half of an O2 molecule. This is correct, but let's look at this process in a little more detail. Here's an actual model of the photosystem II in cyanobacteria. All the blue, purple, let's see, blue, purple, orange, gray, and light blue strips are actually parts of proteins, which are long strands of amino acids. And in simple terms, the chlorophyll in green here, this is chlorophyll, and it's kind of a squiggly lines here, kind of all through the photosystem II, harvests the sunlight, as was shown in the video, which dislodges electrons from the chlorophyll and sends them on their way down the electron transport chain. And the electron first passes through a thiophytin molecule. This was one of the proteins that the narrator talked about in the video. And here's the thiophytin. It actually looks like it's part of the photosystem II, but it's actually dislodged and sent on its way down the electron transport chain through the thylakoid membrane. And after it goes through the thiophytin molecule, it goes successfully to 2 quinone molecules. These are these guys here in yellow, and they're kind of dispersed through the photosystem II, and they also go through the membrane itself from photosystem II to photosystem I. But the main thing I want to point out in this model is the manganese center. That's this thing here, and the water oxidation site here inside the yellow oval. Well, you can't see too much here. This is also known as the oxygen evolving complex, and it's the mechanism that breaks down a water molecule into 2 protons plus oxygen. And this is a model of the oxygen evolving complex, which breaks down a water molecule into 2 protons and half of an oxygen molecule. On the last slide, the oxygen evolving complex was called the manganese center and also the water oxidation site. These terms are all interchangeable, but I will be calling it the oxygen evolving complex, or OEC, since that's a term used in the most recent scientific studies of the complex. It's not yet fully understood exactly how this complex does its job, but I'm going to tell you a bit about some recent work by a team of researchers in Japan who reported their progress in Science magazine in October of 2019. The OEC has been known for several years to consist of a molecule of 4 manganese atoms, a calcium atom, and 5 oxygen atoms, which you can just make out in the center of this diagram. So here's manganese 1, Mn2, Mn3, Mn4. Those are the manganese atoms. And the chemical element manganese, not to be confused with magnesium, which is manganese has an abbreviation of Ma, magnesium, or Mn, sorry. Magnesium has an abbreviation Mg. Manganese has an atomic number of 25, and is located on the periodic table between chromium and iron. It is a required trace element for all known living organisms. However, like many other chemicals, too little of it can kill you, but so can too much. In this model, the 4 manganese atoms are in purple and labeled Mn1 through Mn4. The calcium atom here is in green, and the oxygen atoms are in red and labeled O1 through O5. In addition, the oxygen evolving complex is stabilized by several water molecules. So, for example, here's W3 and W4, and I think there was one more somewhere is W2. Here it is, here's W2. And although I believe there's actually 6 water molecules shown here, only 3 of them are actually labeled. If you look closely, you will see that the OEC is shown having covalent bonds, which are represented by solid lines between the manganese atoms, the calcium atom, and the 5 oxygen atoms. The rest of the structure shown in solid lines is the surrounding protein, known as the manganese stabilizing protein. Again, notice that the OEC is only connected to the water molecules and the protein by dashed and not solid lines. This suggests that the OEC is held in place not by covalent bonding, but by some other force, probably hydrogen bonding. And I'm just going to take a sidelight to point out here that we know how proteins are formed by ribosomes linking amino acids together, but these metal things are not part of amino acids, and so it's not clear to me how this happened. We'll talk about more about that in a moment here, but before we do that, I want to go into the what is known as about this process. And the process that breaks a water molecule into 2 protons and an oxygen atom makes use of the fact that manganese exists in several oxidative states, and the OEC cycles through these states to break the water molecule down into 2 protons, or H+ ions, plus an oxygen ion. The Japanese scientists in the study that I talked about used an X-ray free electron laser to examine the structure of the OEC in these various oxidative states. And so they showed 5 copies of the oxygen evolving complex here in this diagram. And in this diagram, a water molecule here enters the lower right-hand corner in the S0 state. This is the S0 state of the oxygen evolving complex, and it moves clockwise through the S1, S2, S3, and S4 states as the manganese goes from one oxidative state to the next, and before the water molecule is finally broken down. And if you have questions about this process, so do I, and so do the researchers, by the way. Suffice it to say that all the evidence points to the OEC and photosynthesis occurring very early in Earth's history, possibly as far back as 3.8 billion years. Photosynthesis is the main way that oxygen is known to enter the atmosphere. So I want to show this slide, which I showed earlier, and if Hugh Ross is right, as this slide suggests, that photosynthesis and cyanobacteria appeared almost immediately after conditions on early Earth permitted life to exist, the complexity of the OEC, photosystem 2, cyanobacteria, and photosynthesis itself present, I think, a serious problem for the idea that life was started through some process of chemical evolution. Well, there's more evidences of design. Here's a second one. The video we watched earlier showing the basis of how photosynthesis works provided few of the details of the process, and they tend to be complicated. In particular, rubisco was omitted. I think that the narrator said that there was an enzyme that linked the carbon dioxide to the RUBP molecule to form a 6-carbon molecule, which is then broken into 2 3-carbon molecules before being converted into glucose. Well, that's done with this protein called rubisco, which is an enzyme in the first step of the photosynthetic Calvin cycle. So this is a snapshot from the video, which shows carbon dioxide from the atmosphere about to bond with RUBP, but it doesn't show the rubisco, which actually binds the carbon dioxide to the RUBP. So lest you think that our rubisco is simple, this is a picture of it. It's thought to be the most abundant protein on Earth, being present in all photosynthetic cyanobacteria and plants. Its history began more than 3.5 billion years ago, maybe as early as 3.8. In addition to binding carbon dioxide to the 5-carbon molecule ribulose bisphosphate, or RUBP, forming a 6-carbon molecule, rubisco also splits the 6-carbon molecule into 2 3-carbon molecules. And as the video said, these 3-carbon molecules are then combined to form the circular molecule glucose, which is the energy source for all heterotrophs, that is, organisms that cannot produce their own food. This includes all animals. A single rubisco consists of 16 proteins, 8 large subunits in white and gray, that's these guys here, and they're called chain dimers as well, and 8 small chains in blue and orange red. The large subunits are coated in the chloroplast and the small ones in the nucleus of the plant cell. Here's a 3-dimensional image of rubisco, just to show that it was not a 2-dimensional object like you might have thought in the previous diagram. Scientists often cite the enzyme rubisco as a bad design, likely a result of evolution and not worthy of a divine creator. The problem is that rubisco sometimes confuses oxygen molecules for carbon dioxide molecules. And this reduces the efficiency of photosynthesis and produces toxic substances which must be dealt with by the cell's machinery. This process is known as photorespiration. If the cycle operates as it was intended to, apparently, we have photosynthesis, but if instead of carbon dioxide, rubisco converts oxygen, that's called photorespiration. So here's a simplified diagram of the Calvin cycle showing where rubisco fits in at the top of the cycle. And in this depiction here, it takes the RUBP and the CO2 and combines them, fixes the carbon with 6 ATPs, actually 3 carbon dioxides, reduces them to the three carbon molecules, and then forms them into the 6 carbon molecules, and then into sugar, and regenerates the RUBP, and this cycle goes on and on and on. But for every 3 molecules of carbon dioxide that rubisco fixes into a 6-carbon molecule, it also confuses and fixes a 4th molecule of oxygen instead of carbon dioxide. This makes some sense because there's a lot of physical similarity between the oxygen and carbon dioxide molecules. And instead of photosynthesis, this process is called photorespiration. The net effect of this mistake is that no sugar or ATP is produced, undesirable chemicals are produced, and extra energy is consumed in the cell to eliminate them, and the production of glucose is slowed. When rubisco was first invented, notice the term invented. Evolutionists even use that term. Wow. I don't think evolution invented anything, but that's my prejudice, I guess. When rubisco was first invented billions of years ago, carbon dioxide levels in the atmosphere far exceeded that of molecular oxygen. Whereas atmospheric oxygen levels currently exceed carbon dioxide by about 500 to 1, in those days the situation was reversed, so it would have been very unusual for rubisco to have fixed a molecule of oxygen. It should also be apparent that despite these mistakes, rubisco is still very selective, because when presented with 500 molecules of oxygen, as in the current atmosphere, to only 1 of carbon dioxide, rubisco still chooses the carbon dioxide molecule 3 out of 4 times. even though the oxygen is much more prevalent. But is there a consensus that Rubisco is poorly designed? Biochemists who studied it in 2006 said, despite appearing sluggish and confused, most Rubiscos, and actually there's a number of different versions of Rubiscos depending on what photosynthetic process we're talking about. Most Rubiscos may be near optimally adapted for their different gaseous and thermal environments. A Danish team says the efficiency of Rubisco has no bearing on crop yields. Increasing the efficiency of Rubisco would decrease, not increase, crop yields. And finally, according to the biology textbook that I used when I took biology back in 2009, says, we now know that at least in some cases, photorespiration plays a protective role in plants. Plants that are impaired in their ability to carry out photorespiration due to defective genes are more susceptible to damage induced by excess light. Researchers consider this clear evidence that photorespiration acts to neutralize the otherwise damaging products of the light reactions, which build up when a low carbon dioxide concentration limits the progress of the Calvin cycle. Whether there are other benefits of photorespiration is still unknown. More Rubisco facts. Rubisco is thought to be the most abundant protein on Earth. I think I've already mentioned that. It's rich in the most important amino acids, and many people consider it to be a large potential source of food protein. And it's ideally suited to the purposes for which it was created, and is yet another reason to believe in the creator God of the Bible. A third example of an evidence for design is metalloproteins. So what are metalloproteins? They're proteins which contain at least one metal ion cofactor. And of course, the most obvious example of that we've already discussed is the oxygen evolving complex with the manganese and calcium in it. About half of all proteins are in this category. These metals have many different functions, an important one being that many metals can be used to catalyze or speed up certain chemical reactions. We've already touched on the fact that photosystem 2 is a metalloprotein because it contains the oxygen evolving complex with metals calcium and manganese, the active site of which is responsible for breaking down water molecules into protons and oxygen. So here's a few other metalloproteins. Chlorophyll. Well, we've already talked about that. Photosystem 2 also contains chlorophyll, which contains a magnesium ion. And here's what the molecule of chlorophyll actually looks like. You see the magnesium in the center of it attached to these four 4 carbon and a nitrogen circles, which are encircling the magnesium. These 4 rings are called pyro rings. There are several different types of chlorophyll occurring in both cyanobacteria and chloroplasts, and all have a similar structure. Don't worry about the details. My point is that there's a magnesium ion at the center. And a third example, we could list many others, but I'll stop by listing Rubisco. Yeah, the one we just talked about, the enzyme that converts carbon dioxide into glucose. Here's a picture of the active site of Rubisco. The green sphere in the center is another magnesium ion. The magnesium binds the ribulose bisphosphate to the carbon dioxide molecule that will be attached to the 6-carbon molecule. Many other metals are also included in other metalloproteins, iron, for example, cobalt, nickel, copper, and zinc. And a final example of the evidence of design. Well, we haven't got to that yet. Let's just talk about the problems with metalloproteins first. It might not sound out of the ordinary, but there are several problems relating to metalloproteins. First, we know that all proteins are made when ribosomes assemble acids according to the genetic code. Metalloproteins are similarly made by ribosomes, but the amino acids don't contain metals. I haven't been able to find much information on metalloproteins at all on the internet, or in my biology textbooks, the addition of a particular metal ion is necessary and does not appear to be random. The oxygen evolving complex always includes a molecule containing 4 manganese, 1 calcium, and 5 oxygen atoms. Chlorophyll and rubisco always contain a magnesium atom. Most likely, these metals are incorporated into the finished protein during the folding process. But how? I theorize there must be a reasonable process. I just haven't been able to find out what it is. There's definitely elegance and complexity in it. Finally, at least in photosynthesis, metalloproteins appear to go back as far as when oxygen first appeared in the atmosphere, probably more than 3.5 billion years ago. The elegance and early appearance suggest to me that an evolutionary explanation is extremely unlikely. It seems to me we're seeing the work of a designer, the creator of the universe. Finally, here's one final example revealing that the process of photosynthesis appears to be designed. Recall that earlier I said that some chloroplast proteins are formed from the chloroplast DNA, but the majority are actually formed from the nuclear DNA manufactured outside the chloroplast and then transferred into the chloroplast. In fact, remember that I said that the rubisco molecule, the one that converts carbon dioxide into sugar, consists of 8 large subunits, which are made in the chloroplast, and 8 small subunits, which originate in the nuclear DNA outside the chloroplast. This diagram shows roughly how the nuclear proteins are transported into the chloroplast stroma and the thylakoid. So here we're in the cytosol of the cell. Here's the chloroplast outer membrane and inner membrane, the stroma of the chloroplast, and one of the thylakoids. Remember, they're stacks like coins. The thylakoids are stacked like coins. At the top of this diagram, in the cytosol, you see 2 nuclear proteins, the red one and the green one, squiggly lines, being escorted to the chloroplast by stromal transit peptide sequences. These are the yellow bars. These pass through the chloroplast outer membrane through the TOC protein. We haven't discussed that one yet, have we? And we're not going to. And the TIC protein. Trust me, they're made somewhere. I think they're probably made in the nuclear DNA, but we don't know. I don't know for sure. It kind of looks like they don't go through the TOC protein the way the diagram is shown, but they really do. They go through both TOC and TIC and into the stroma. And once inside the chloroplast, the yellow stromal transit peptide sequences are no longer needed, so they're removed. We see them removed. Here's the red. But now we see that there's a blue transit protein. That's called the thylakoid transit peptide sequence, and it enables the protein to enter the thylakoid, which is the oval at the bottom of the picture. SRP stands for signal recognition particle. TAT stands for twin arginine translocase. SEC is a secretory protein, which permits the thylakoid to secrete waste material. Interestingly, these 3 pathways, SRP, TAT, and SEC, and those are not the only proteins that have to go into the thylakoid, obviously, because we've got photosystem 2, and we've got chloroplasts, and not chloroplasts, but chlorophyll, and we've got the oxygen evolving complex. And but anyway, interestingly, I think these 3 pathways, SRP, TAT, and SEC, are powered by 3 different methods that are shown here. ATP in the case of SEC, the proton gradient shown here by delta pH, and a combination of GTP, which is guanosine triphosphate, and the proton gradient in the case of SRP. I think it takes a lot of faith to believe that evolutionary processes came up with all this essential stuff. Let's think for a moment the implications of this for a protein complex like rubisco. First of all, remember that each leaf cell contains 100 or so chloroplasts, and here we're looking at just 1. When more rubisco is required, the chloroplast DNA makes the large subunits, and the cell's nuclear DNA makes the small subunits. Think of the coordination that must be required. The nuclear DNA provides small subunits for 100 chloroplasts, but the chloroplast DNA only provides large subunits for itself. These units must all come together at about the same time or the Rubisco will be incomplete. How this is done is beyond the scope of this lecture and beyond the scope of my brain, but I think it's obvious a lot of coordination is required, suggesting to me that the process is exquisitely designed. I close with this quote from A.J. Roberts, biochemist and former RTB scholar and virologist who spoke to our chapter earlier this year. In one of the papers she wrote on the RTB website, she said, "As I consider the underlying complexity of photosynthesis, which is the most basic process of energy harvesting in Earth's ecosystems, the competing worldview of Darwinian naturalism seems much less persuasive. The amazing nature of trees and other photosynthetic plants that produce far more food resources than they themselves need, while serendipitously providing oxygen as an unneeded byproduct for sustaining all animal life, is one such example that fits nicely in the biblical worldview of God's extravagant provision and progressive creation of finely balanced ecosystems. Meanwhile, naturalism's non-teleological selection of the fittest seems a truly incredulous claim to account for such interdependent, highly complex phenomena." Thank you, and I'll be happy to entertain any questions that you might have.
I have a question.
Okay.
What's your background to know all this stuff?
Okay, great question. I'm an electronics engineer by trade. I got very interested in biology in 2004 when I saw an article or an editorial in the San Jose Mercury News written by a professor of theology at Santa Clara University, which basically said, those of you who don't believe in evolution need to get over it. Well, I didn't like that, so I wrote my own editorial to the San Jose Mercury News. Since I don't have a PhD in anything, much less biology, it was ignored. So I decided that I was going to go back to school and learn all I could about biology, well, without putting myself out too much, because I went up to Foothill College, which I think is excellent, by the way. If you ever have an opportunity to go to Foothill, I suggest you do it. And I took general chemistry again because I hadn't taken it in 50 years. I thought it might be useful to spiffy myself up. Then I took their biology course, which is a one-year course, and it's used for pre-nurses and pharmacists and doctors. It's a heavy-duty biology course using a heavy-duty book. And then I took organic chemistry. And then I started reading a lot of books, RTB books. That's how I got involved with RTB. A lot of intelligent design books, and just a lot of general biology books. So it's basically a lot of study that I've done. So no, I don't have a PhD in biology. Any other questions? Don't hesitate to ask them. And as Hugh Ross says, I like your hardball questions too, not just your softball ones.
Hey, Kirby, you remember me? I met you like 3 years ago. It was a long time ago.
Yeah.
I live way out in Manteca, so I don't have a chance to come into the Bay Area too often. But have you heard of Perry Marshall? He's got a book called Evolution 2.0. And
I have, and he's actually sponsoring a conference that goes on next week, and I've signed up for it. It has more to do with cancer than it does with evolution, but he's talking on it, and a number of heavyweights, Dennis Noble and can't think of the guy's name.
Yeah, I know, and he's got
but some heavyweight players are George Church, I guess, is one,
that's right,
and there's a number of others. but yes, I have heard of Perry Marshall. I haven't finished reading his book. I'm still reading it, so I can't speak very knowledgeably about it.
Oh, that's he's got some. It's a new version of evolution with it's not so much incremental. It's more giant leaps of recombination and other
Yeah.
intelligent-based cells. And he's a believer. He's a Christian. He's just a different take on evolution. So I
Okay. Yeah.
just
Well, I don't claim that my view of evolution is the right one. I know that there are a lot of Christians that believe in theological, theological evolution, I guess it's called, or
yeah.
creationary evolutionism or something. But it's basically they believe in full-fledged evolution, everything, But they also are quite faithful in their reading of the Bible, and they figured out a way to put this all together. I don't really have any problem with that idea. I just can't see the science. There's too many holes in the science, I think, to make me believe in macroevolution anyway. I believe in microevolution, microbial evolution, and even speciation, but I don't believe in chemical evolution or macroevolution.
I'm on the same page with you exactly.
Okay.
Yeah, actually, just a comment.
Okay.
Marshall was talking to Stephen Meyer, and or someone was meeting between the two, and I think Meyer's question was all these fast processes that David was mentioning, because I read 2.0 as well, and Stephen Meyer asked the question, same question in the back of my mind, where all these fast mechanisms come from. So, I mean, yeah, it shows that these things can advance fast, but where did these mechanisms come from? So
yeah, and the intercoordination between them, like
right. So there's
yes.
Yeah.
Having not read the book, I better not comment on it.
No, same here. I've not read it either. I need to.
But I am reading it, actually. I'm starting to read it. So,
okay.
So I'm not sure that I will be able to answer it even after I ask questions. But I want to point out that Perry Marshall is not a biologist either. He also is an electrical engineer, like I am.
My son put a question in the chat.
Your son.
Kirby thinks that Foothill professors are great, and he graduated from Harvard. This is way more. Have you heard of? Okay, how long does photosynthesis take, and does the time vary when it comes to different plants? Great question. That's really hard. It's when you say how long does it take, remember that there's the light dependent and the light independent reactions, and and there's 100 chloroplasts in each cell, and there's maybe 1 million cells. There's 1 million or maybe 2 or 3 million cells in each leaf. So it's going on kind of continuously, and it doesn't take very long for the electron transport chain to work its magic, and for the proton gradient to work up and to start pumping hydrogen molecules through the ATP synthase and creating the ATP and the NADH and breaking down the H2O, and then starting the Calvin cycle. I would guess it's probably no more than a second or two between each glucose molecule that's produced. And I actually don't really know the answer to that question. It's pretty fast, but I'm not sure exactly how fast. Good question, though.
So regarding how photosynthesis somehow started as soon as the rocks were no longer molten, is there any explanation for that? That looks totally
well. Yeah, that's a great question too. So my explanation basically, and it's kind of the same explanation as how life first got started on Earth, and that is that chemical evolution does not have a good explanation yet. As I think Jim Tour has said many times, maybe in 100 years or so they'll have some idea, some clue, but as of now they really have no clue. They have some arm waving ideas, but whether it's driven by replication first, or metabolism first, or membranes first, they really can't say, and so my best explanation at this point is God did it. God somehow created the first life forms. We haven't come up with a better explanation than that. And that would include both the first metabolism forming. Well, basically, when life form was created, that meant that some metabolism formed. Now, maybe it took a little while for the first photosynthetic cell to be created, but I don't see any way that, especially as complicated as photosynthesis is, and especially as complicated as all these proteins that existed obviously long before the first eukaryotic cells first existed. It seems to me like this is really kind of beyond the scope of any evolutionary processes. That's my answer. And maybe I'll eat my words in 100 years or so if I were still here. Go ahead, John. Do you have a question? Or you just...
Yeah, no, I have a sort of a comment and a question. No, not a question, but a comment or a clarification. Well, first of all, I just want to congratulate you on putting together this very interesting talk. And I'm sure it took a lot of work on your part and tenacity. So I really appreciate it. And I think to those of us who don't really have a lot of biology background, but your fundamental thesis is that this complicated photosynthesis process appears to exist 3.85 billion years ago. 3.85 billion years ago.
Well, it's not entirely clear that it was that long ago, but it looks like it was more than 3.5 billion years ago. So
okay, 3.5. All right, close
more
enough.
More than 3.5. Yeah.
And then you, so it's a very complicated mechanism, and you showed some of the intricacies of the handshakes be among all the subcomponents necessary for this photosynthetic process to take place. So, so number one, it's a very complicated machine, and then you mentioned the watchmaker's argument.
Yes.
So where did this complicated machine come from, right? Because so you invoke the watchmaker, so some intelligence had to put together this thing. Or the other argument is that, well, maybe it evolved, but then how could it evolve when all of these different components that are operating in a sophisticated programming sequence? You have to do step A, step B, step C. Who's telling it to go through that sequence? Or is it once you start step A and then step B, C, D, E, F follows naturally? We don't know, right? So where is that intelligence coming from? Where's that information coming from? And so then you question, how can an evolutionary process come about so early? And how can an evolutionary process program in that intelligent programming? Insert the programming into the photosynthetic process, unless someone had designed it to begin with. So that's the bottom line, right? For us Christians to use the marvel of this process.
Yeah, and just because, there are a number of steps that aren't really explained yet, and that coordination step is a big one. There's also, of course, going back to the origin of life, there's also, what about the homochirality problem? What about how did the genetic code come about in the first place? And yet those things had to have been in place right from the beginning, because that's what we see throughout life now. And it's not just in eukaryotic organisms, it's also in all the prokaryotic organisms. So yeah, John, you got a good point.
Yeah, and I just thought of another sort of a consequence of this mechanism is that this basic mechanism is still the same mechanism today.
Yeah,
right?
right.
It hasn't changed, has it?
Yeah, and so this Rubisco, which might have worked better 3.5 billion years ago when there wasn't much oxygen in the atmosphere, is still around today. So, and you know, and now they're thinking, well, maybe it isn't such a bad design after all.
Yeah. So you would think that in 3.5 billion years, this process, if there is some learning from natural selection, would have changed this process.
Yeah.
But then, if the process is still the same, that means nothing basically has changed.
Well,
so where's the evolution? I don't see the evolution in this process. Do you?
Well, okay. So there is a big difference between eukaryotic organisms and multi-cell, especially multi-celled eukaryotic organisms, and these single-celled prokaryotes like cyanobacteria and alpha protobacteria. And the multi-celled eukaryotic organisms didn't really start even existing before 600 million years ago. So there was basically almost 3 million years of, well, actually 2.5 million years of just the single-celled prokaryotic organisms. Well, actually, the single-celled eukaryotes were supposed to have started about 2 billion years ago. So it's not entirely true that but that was only single-celled eukaryotes. And so anyway, single-celled prokaryotes, 3.85 billion years ago. First eukaryotes about 2 billion years ago, and the first multi-celled eukaryotes about 600 million years, well, actually 540 million years ago, I'm going to say. Even though, you know, there was some indication of some multi-celled organisms before in the Ediacaran era, so before the Cambrian. I was kind of expecting somebody to say, you know, that graph that Hugh Ross showed of atmospheric oxygen. First of all, don't we have a lot more than 10% oxygen right now? Yeah, we have about 20%. That didn't show the last 10% being added. And what was that lump in the middle at about 2 billion years ago? Well, that was called the Great Oxygenation Event. And that occurred at about the same time as the continents began to rise out of the water. And actually both photosynthesis and methanogenesis were combining to pump both oxygen and methane into the atmosphere. And this was actually using up more and more water. And most of the oxygen was actually being formed into was being used to oxygenate the elements, primarily the metals. And anyway, that's what the lump was, although it's not really well known as to why it appeared as a lump. But apparently after the Great Oxygenation Event, oxygen levels rose to about 1% and remained there until they rose to about 10% in about 600 million years ago. So, oh well, that was a question you didn't ask, but so I won't answer that one. So why did it take God so long to get from the single-celled organism to the more complex stuff? Seems like he took a while to figure it out, which doesn't seem right. Was he learning as he went along? Don't mean to be heretical. I love Karen's answer to this, and I'm going to ask Karen to step in and ask why it took God so long to do all this stuff. You're muted, Karen, so unmute yourself.
I couldn't help but think that, wow, our God modeled love and patience, patient love in an intimate way by enjoying what he was doing so much as he created, step by step. Yes, he could have done it in a snap of a finger, but why would he do that when he loved so much? When you really, truly love, you take your time and you do it from your loved one's perspective.
I love that. I love that answer. Unfortunately, there actually also is a scientific answer, and that one's explained in Hugh Ross's book, several of them, Hugh Ross's book, but specifically in Improbable Planet. And it took that long for the various photosynthetic cells to build up oxygen to the level where, so at 1%, that wasn't really enough to support to support eukaryotic multi-celled organisms such as well, even fish, but certainly not us. And so and it wasn't just the oxygen, it was a whole series of other things that had to come together in order to prepare the Earth for advanced civilization. And you can get into that more in Hugh Ross's book, Improbable Planet. So let's pray, and then we can stick around. I will stick around as long as you all stick around. Lord God, thank you so much for the opportunity that we've had to meet together. We don't know what's coming up with this coronavirus, so we do ask for your protection for the coronavirus workers and for those who are infected by it, for healing for many. And we know that you are in control of the whole universe, much less this little speck of a planet called Earth, and the troubles that we all have individually are minuscule compared with your power and glory. And we thank you, Lord, for your power and glory and for the miracles that we see around us. And we ask for your blessing now on us as we conclude this meeting. And we pray these things in Jesus' name.